Well, thank you all for coming. Those of you who are attending virtually, thank you for coming over the internet. When Mark asked me to talk at this symposium, I said to him, Mark, I don't work on coral interactions. And he said, well, that's all right. He said, I know you don't do it that much anymore anyway, but tell us what you think. So what I'd like to do is very briefly set the stage for what I think are the important issues. But I don't want to go into too much detail about the nitty-gritty of coral-algal interactions, because you're going to have a whole day of that from all the other speakers, and I would really just be repeating much of what they have to say. So what I'd like to talk about to start off with, and in a way it might have been better to do this at the end than we'll do it at the beginning, I'd like to talk about what we don't know about the consequences of coral-algal interactions rather than the mechanisms through which corals and algae themselves interact. So, but as I said to Mark, although it's not something I do a lot of active research on, really ever, and certainly not now, it is true that it's a story that's very near and dear to my heart, at least near to my heart, maybe not dear, and that's because I witnessed early on in my scientific career the kind of quintessential example of what happens when corals and algae interact to the detriment of the corals. And this was a story, many of you are way too young to have actually ever seen a reef the way it's supposed to look. Actually, even this picture of a reef taken in 1975 when I was a beginning graduate student isn't really the way a reef should look. And that's because if you look up, can you see, can we dim the lights a little bit?
Yeah, we're going to try to get the lights down in the front.
So, this was a picture taken at Discovery Bay, Jamaica, and those of you who are not used to looking at coral reefs, corals on them, everything you can see here is living corals and branching corals, Aquaphora cervicornis, and this is a bushy coral, Dracis, and head corals. And these reefs were literally 70% living coral when those of us who were there at the time started working. Now, we knew those reefs were not in pristine condition, and you can tell that by what's not in this picture, namely any kind of big fish. So, you see a few fish up here in the upper right-hand corner. Those are all plankivores. They're only about this long. So all the big fish were already gone, even by the 1970s, because Jamaica is a very poor country, and essentially poor people need to capture fish in order to feed their families in this part of the world. So the fish were largely gone, but the corals were still there. There were a lot of graduate students and faculty members, current and future leaders in coral reef biology. And I have to say, we'd sit around talking about the reefs, we'd talk about them as like overfished, but none of us ever worried about the corals themselves at that period of time. We took those corals, or the coral reefs, for granted because we never made any kind of connection between the absence of fish and any other environmental things that might be going on and the health of the corals. Now that turned out to be a big mistake because within about 6 years of this picture being taken, those coral reefs were essentially gone. They became completely dominated by seaweeds, which you can see here. And it was a dramatic and very rapid transition, which I witnessed essentially during my first years as an assistant professor. And it really, when you see an ecosystem that you take for granted essentially vanish before your eyes, it does something to your scientific perspective in terms of what is important to study and the appropriate time and spatial scales for which you need to study it. And this was that kind of a dramatic event. Now, of course, for those of us in Jamaica, we saw it firsthand because there was a major marine lab there, the Discovery Bay Marine Lab, but this was not a phenomenon that was limited to Jamaica, and I'm not going to go into the details of all the things that contributed to the demise of corals in Jamaica and throughout the Caribbean. But here you see a meta-analysis that was done, and this is coral percent cover here, 1977, essentially the same time this picture was taken, 2002, and you can see the steady decline of living coral cover. And in fact, there's been an 80% loss of living coral in the Caribbean over the 30 years that the study was done. And that kind of decline certainly rivals anything seen in most tropical rainforests, for example. So this is really serious environmental catastrophe. And it's not even necessarily limited to the Caribbean. And in fact, a slightly more recent meta-analysis done, this one was done by Gardner and colleagues, and the more recent one on the Pacific, which I'm not showing the data for, by Bruno and Selig, showed that the average percent cover, living coral cover in the Pacific was about 20 to 25%, so about here. So essentially you can say that Pacific reefs are about here in this trajectory. Healthy coral reefs really should have 50, 60, 70, 80, sometimes they even have 100% living coral. But reefs around the world are approaching this level of 5, 10, 15, 20, 25% cover in most places around the world. And perhaps not surprisingly, even more recent analysis suggested that about 1/3 of all coral species are at risk of extinction due to what's going on in coral reefs globally. Now, why is this happening in a very big picture sense? This is not the detailed interactions that you'll be hearing about throughout the rest of the course of this symposium, but it's essentially because we're doing this giant experiment on planet ocean, and we're putting a lot of stuff into the global ocean, we're taking a lot of stuff out. So on the input side, we're putting in nutrients, which I'm sure some of the speakers will talk about the role that nutrients have in stimulating the growth of seaweeds, which are the primary competitors of corals, which is why this symposium is about coral level interactions, toxic materials, sediments, invasive species, which in the case of the Caribbean, we now have an invasive fish, the lionfish, which is eating all the few fish that remain, so even aggravating the problem of this coral algal imbalance. And of course, carbon dioxide, which both warms seawater and kills corals because they're very sensitive to small increases in temperature, but also by dissolving in seawater is making seawater more acidic, and that also tends to shift the balance against corals and in favor of seaweeds. And then on the output side, we're essentially taking out anything big, including all the big herbivores that eat seaweeds. So you have a situation where corals and seaweeds are competing for space, and almost everything that people are doing to the planet essentially changes that competitive balance to the detriment of corals and in favor of seaweeds. And that's why we're seeing, in a very broad brush sort of way, why we're seeing the changes that we've seen. Now, how do we typically describe what's going on coral reefs? Well, these are a couple of examples. This is the now iconic paper by Kerry Hughes, where he described in detail what happened to the reefs of the north coast of Jamaica. And here you see coral cover, again, you can see, depending on the depth, ranging from 7 to 35 meters, anywhere from about 50 to about 70% living coral. In 1975, by 1995, it was less than 10%. And conversely, you have algal cover increasing over that same period. And then a more recent study by Stuart Sandin and colleagues, including Jen Smith, who will be speaking later today, we see again a description of benthic cover as a function of coral, crystallose coral and algae, turf algae, macroalgae, and others. And this was I'm sure she's going to be talking about this in great detail, so I won't belabor this point, simply to say that this is this is a gradient of human disturbance, where this is the most disturbed and this is the least disturbed. And so you see coral declining across that gradient, and macroalgae becoming more important in the most disturbed setting. So that's typically what we do when we describe this transition, and often, well, not always, parallel to this are the descriptions of the fish community, typically represented as biomass, either overall or as a function of specific trophic categories. Now, what about the rest of the benthic community? And that's really what I want to talk about now. And really, the bottom line is we don't really know what's happening to the rest of the benthic community, apart from the corals and the seaweeds. But we're finally getting the tools that will allow us to find out. So I'd like to tell you a little bit about this kind of work. So This is a picture of the undersurface of a living coral. And as you can see, it's covered with lots of different kinds of living organisms. With sponges and bryozoans and little bits of algae, and of course there are lots of small mobile invertebrates scurrying around. You put something like this under a microscope. And it's been estimated that there's somewhere between 1 and 9 million species that live on coral reefs in total. Now, this means that the diversity of these associates of coral reefs far exceed the diversity of corals. There are only about 1,000 species of scleractinian corals described, and although the fish diversity is higher, it's certainly not in the millions to 9 million range. And this means that the bulk of the diversity of coral reefs, which are mega diverse environments, which is why they're called the rainforests of the sea, or why rainforests are sometimes called the coral reefs of the land. These communities are incredibly diverse, but essentially unknown. This number of 1 to 9 million species, it comes from two estimates. One is based on an extrapolation of the number of insects in tropical forests, and then making calculations that are associated with the area of coral reefs versus area rainforests. You can imagine it's a pretty tenuous set of assumptions that link those two, the number of insects in rainforests to the number of species of coral reefs. And the other was based on a partial count of everything living in a tropical aquarium in Baltimore, based on about 5 meters squared collections and extrapolated to the global reef environment. So amazingly, actually, there was some overlap between these estimates, but they're still what I would call fairly rough. Now, the other way of thinking about it is to look at groups where we know something more about the diversity, and that primarily is our fishes, which are the best known taxonomically. And it's been estimated that about 1/4 of all the marine species of fishes are associated with coral reefs. Now, so that gives you an idea of the incredible importance of this diversity in terms of our Earth's planetary diversity heritage. The other thing that's worth noting is that almost all of these species, actually in the ocean in general, and certainly on coral reefs, almost all of them lack scientific names. They've never been formally described by taxonomists. About 95% of the species in the ocean in general, and on reefs in particular, have never been studied scientifically. Some of them are also really beautiful. Right? So, and in addition to all this huge amount of species, there's the enormous complexity of the interactions. This is a food web that was presented by Bascombe and Enrique Sala and others on a Caribbean food web. And what you see here is the network of interactions. And so each species is shown here as a circle, and then all the lines are the interactions between them. And it's important to recognize that only 30% of the species and 11% of the 3,000 some odd interactions are shown in this diagram. The reason is if you showed them all, it would just be a black circle, so you wouldn't be able to see anything. So they're incredibly complicated communities, and we know given the fact that we know almost nothing about most of the species that live on creek reefs, and just in terms of their name, not surprisingly, we know even less about the interactions. So what do we know then? What can we say, given that sort of state of ignorance, what can we say about what might be happening to the diversity of reefs as you move from a coral-dominated system to an algal-dominated system? And it's remarkable how little information we have on this topic. And I'm going to show you a few graphs that have been published. But this is a topic that has been largely ignored by coral reef biologists. And in fact, if you go to the international coral reef meetings, there's another one coming up this summer in Australia, and you look at the sessions, and there'll be maybe one, there are often 5 days, 11 concurrent sessions, and usually there's 1 or 2 sessions on biodiversity. So there's a trivial amount of effort, research effort being made by the coral reef research community on basic biodiversity. And so as a consequence, there's very little data on what happens to anything other than a coral, seaweed, or fish as you move from coral-dominated to algal-dominated systems. This is an exception to that, which is a paper published by Wilson and colleagues in 2006. And what it shows is that diversity declines with a loss of living coral. So here you see the decline in live coral cover as a function of percentage, and this is a percent change in species richness. And you can see a pretty striking decline, particularly when you have large amounts of coral loss. You get fairly large drops in fish diversity associated with it. But it's also important to realize that you don't always see a loss in diversity when you have corals being partially replaced by seaweeds. And this is work, again, done in the Line Islands in parallel with the work that Jem will be talking about later. And what we did, and I'll a bit later, I'll tell you a little bit more detail about how we went about doing this. This was an estimate of the number of decapod crustaceans, that's shrimps, crabs, and lobsters, living with dead coral in the 4 islands that were visited during the 2005 cruise that she'll be describing. So again, here you have the most disturbed island, Christmas Island, and the most pristine, the one that has actually no people living on it at all. And this is the number of decapod species. And you can see there's actually no difference across those 4, despite the fact there's a pretty big difference in coral cover and algal cover, conversely algal cover among those 4 islands. So across this gradient of human impact and coral loss, there seems to be relatively little impact on diversity, at least for these kinds of organisms. But it's important to realize that it's not just coral cover that's being lost. It's also this phenomenon that's been described as reef flattening. And reef flattening occurs because coral reefs are like cities. They're a constant and dynamic replacement of some corals growing and then other corals dying, sort of like being in having a race between a construction crane and a wrecking ball, where the things that chew on dead coral skeletons, like sea urchins and fishes and other boring organisms, are gradually turning dead coral into sand at the same time as new coral skeletons being created by living corals. And if corals are losing the race, if you will, in a wrecking ball construction crane context, then essentially these reefs turn into sand because the new growth doesn't keep up with the bioerosion. And here you see a really vivid example of that in reefs, pictures of reefs that were taken from Barbados from the air in 1950 and 1991. This is the exact same reef, so you can see the same sort of profile, and these numbers and dots show the analogous points between these 2 time periods. And you can see that there's just a lot. See, this whole part of the reef, places 2 and 3, is gone, and here's 4, and here's 8, it's gone. These reefs are physically disappearing, not just living coral, but the reef structure itself is disappearing. And in this particular reef, there was about nearly a 20% loss in actual reef rock, which was converted into sand. And this has been, as I said, described by flattening as the flattening of reefs. And really nice study by Alvarez Philip in 2009 did, again, a meta-analysis looking at how the 3-dimensionality or the rugosity of coral reefs has changed over the same time period that we've been talking about for these other changes, that is the 1970s essentially to the present. And they divided rugosity into 5 categories. Rugosity is measured by laying can be measured by laying a chain over the reef and looking at the ratio between the total length of the chain, which reflects that 3-dimensionality, versus the length the chain would be if it was just a straight line. So if there's no difference, if the reef is flat, then you have a rugosity of 1, and it can get higher and higher, up in principle to infinity, although in practice, rugosities above 3 are relatively uncommon. So here you have these 5 categories. So this is 1 to 1.49, 1.5 to 1.9, and up to this darkest color, greater than 3. Here it is in the 1970s, 80s, 90s, and 2000s. That's a really dramatic change in the 3-dimensionality of reefs over a relatively short period of time. We essentially lost all the really complex reefs in terms of the 3-dimensional structure. And most of these, look at this. This is 60% of the reefs are basically flat. That's really quite a striking change in terms of what's happened to the 3-dimensional complexity in the region. Now we know that this 3-dimensional complexity, again from a relatively limited number of studies, is pretty important in terms of biodiversity. The same paper, this Wilson et al. Paper that I showed you earlier about the loss in diversity of fish as a function of the loss in living coral cover, also compared the losses in terms of whether the losses were due to physical events such as typhoons and hurricanes, or biological events such as coral bleaching or outbreaks of crown-of-thorn starfish. And the reason that distinction is important is that a crown-of-thorn starfish or bleaching will kill the living coral that leave it until, as they say, the reef turns into sand. But immediately after that event, the three-dimensional structure of the reef will still be present. It's just there'll be no living coral. Whereas in a physical disturbance event, you're in addition to killing a lot of living coral, you're also eliminating the three-dimensional structure. And you can see that in these are different kinds of fish. Obviously, fish that feed on corals are most affected, and things that are obligately living with corals, the second most affected. But in every case, for all these different kinds of trophic categories among the fish community, the physical disturbance always had a much bigger impact than biological disturbance, which suggests that if things don't recover from the biological disturbance, and those reefs essentially do ultimately turn to sand, the losses that are being reported now are actually just a shadow of what may ultimately occur as these now dead reefs are eroded by fishes and urchins and other organisms. And the most striking relationship between topographic complexity and species diversity, actually in this case generic diversity, that I know was published by Jody and Peter Edmonds in 2006. So this is a measure, excuse me, of topographic complexity versus the estimated invertebrate generic richness. And this is in the Caribbean. You can see a really nice relationship. They also plotted the relationship between invertebrate generic richness and percent living coral, and the relationship was much weaker. So this is actually the strongest relation with rugosity. But the thing I want you to notice is the total. Look at the variation. They're working between about, say, 15 and 35 generally. That is a tiny, tiny fraction of the total generic diversity that's typical of reefs. So they're looking at just the big things that we can easily see and count. This is, of course, not Caribbean, but this gives you a feeling of how lonely it can feel to be an invertebrate on a flat reef. So, what I'd like to do now is step back from this question of what we might be losing, because there's not much more data than what I just showed you, and talk to you a little bit about just how we estimate on what's on reefs at all. And this is a cartoon by Jim Toomey, which was done for this in honor of the Census of Marine Life, which does, I think, also inadvertent. He's a big sharks. Sherman's Lagoon is the name of the cartoon, as opposed to all cartoons. But it does also sort of inadvertently also illustrate that we know a lot more about the big things than the small things, because what he's counting up here is rays and great whites and white tips and mangrove sharks. So, the reason we know so little bit about invertebrate diversity is basically that the numbers are so high, nothing has any names, and the number of people like me that actually knows how to identify something is not a fish or a coral, pretty small relative to the total number of things there are. And so what's needed are new methods that really allow us to standardize assessments of diversity, sort of quasi-automate those assessments, and scale them up so they can be done globally. And the way that we're approaching this, and this is a project that was started under the census, is a combination of using standardized volumes or surfaces. This is a deadhead of coral. This is something called an autonomous reef monitoring structure, or ARMS, which we deploy on the reef. It has lots of layers. Leave it there for a year and take it back, and you can quantify both the sessile things that live on it and all the pre-corals that crawl in between the layers, and of course combine it with DNA, because if nothing has names, there's no point trying to identify it using a taxonomic key. It's much quicker just to use genes to give you a sense of how many species. We use genetic barcoding primarily up until this point, although at the very end I'll talk to you a little bit about some work with using next-generation sequencing. So we have these studies in a lot of different places. These ARMS have been deployed everywhere you see red and orange dots. And I'll talk about the data from these 7 sites around Australia and the Central Pacific and the Caribbean. But before I show you the data, I really want to emphasize how much more efficient these mechanisms are than traditional biodiversity surveys. Now, Philippe Boucher is probably the best all-taxobiotic inventories person on the planet, that's ATBIs. And he and colleagues went to the Philippines. I think something like 75 scientists spent about a month each. It's equivalent to 6-person years. And in the context of that, they found 1,200 species of decapods. And using sort of standard biodiversity grades, you can estimate that there probably are about 900, using the same methods and the same level of effort, you find about 900 species in the Great Barrier Reef. Well, we found 205 species in just 2.1 meters square of deadheads and these artificial surfaces. And it took us just a couple of weeks to do it. And similarly, we have, bringing our class of students for 2 days to Indonesia, we found about 400 species of decapods. So you can find about 25 to 35% of everything that's there using these methods at a fraction of the effort. And that means you can do these kinds of studies all around the planet. Philippe has only done it in about 3 or 4 places in his whole life because it's so time-consuming. So to give you a feeling for the kind of diversity you get, this is a study we did, as I said, in those 7 sites, and we found that in just a little over 6 meters squared of dead coral heads, we got the equivalent of about 80% of the entire crab diversity of all of Europe. So it's encouraging that we can get these numbers. It does tell you that the scale of problem is so hard, that it's so large that it's still a long way to go in terms of really understanding what's living there. These are what the data tend to look like when you plot them as rarefaction curves in terms of the number of things that we sample versus the number of species. The species are distinguished by the cytochrome oxidase 1 gene. Anything that's 5% different or more is called a different species. And what you can see is the numbers keep going up. That means we haven't come close to sampling the entire community. And only about 5% of the sequences are even in GenBank. So a lot of these things are just unknown. And so given that, you say, okay, well, this is what we've done so far. It took us 4 or 5 years to do this. How many species might there be altogether of reef brachyurid crabs? And we chose that because brachyurid crabs, which are the sort of true crabs, are actually the best known of all the marine invertebrates apart from the mollusks. And it depends on doing one of these species area relationships. Species equals area raised to z. The area sampled 6.3 meters squared. It's not very much. Total area of coral reefs is 600,000 square kilometers. And the answer you get depends entirely, not surprisingly, on z. And so people typically use z values, often of 0.25, with the range between about 0.1 and 0.4. But because it's an exponential relationship, it has a huge impact on your estimate. So if you have z 0.1, then the total number of reef brachyurids is probably about half of all the described reef brachyurids. Whereas if z is 0.4, the total number of reef brachyurids is about 800 times the total number of brachyurids described today. So you can see there's still a huge amount of variation because of the difficulty of doing the sampling. And that leads me to my last couple of slides, which has to do with next-generation sequencing. Genetic barcoding is a lot faster than sending thousands of specimens off to taxonomics experts and then waiting 5 years to get an answer back as to what you might have sent them. But because there are so few of them and so many things to identify, so it's a lot faster, but it's still fairly tedious. You have to take a little piece of a leg off of every single crab and put it in a little vial and individually amplify it, sequence it. Much better is to take one of those arms and a chisel or a paint scraper and scrape the entire thing into a jar of alcohol, which is what you see here, and then amplify. You don't amplify everything because it becomes so complicated bioinformatically to figure out what you've got. So you amplify based on primers. Again, we use CO1, cytochrome oxidase 1, or sometimes some of the ribosomal genes, and then census the DNA sequences all at once using these next-generation sequencing approaches. And so I just want to show, this is you'll be hearing work on microbes. People working on what microbes have done this for a long time, and it works very well. It's actually harder to do it, strangely enough, with larger organisms because they have they vary so much in their size. It's a little hard to know if what you put in is actually what you're getting out. But to give you a feeling, this is literally off the sequencer in the last week or so. And so these are 50,000 out here, numbers of individual sequences, and this is the number of species. And as you can see, the graphs still look the same. But I think with this method, we actually have some hope for, this was just 3 arms that we analyzed because we wanted to see if it was really working. And we're getting hundreds and hundreds of species of a variety of different phyla, really across the phylogenetic spectrum. And what's really interesting is this was done from Maria. Some of you may have heard of the Maria Biocode project, which has involved 5 years of intensive effort to get a barcode for every single organism on the island of Maria. And despite that 5 years effort, about 30 to 50% of the sequences we're getting here have never been picked up by the biocode effort. And this is just 3 little pieces of samples. So it shows how much there still is to do. And so as a consequence, I'd like to end with this, which is sort of our current best estimates to what we might be losing as corals are being replaced by seaweeds and are flattened, in particular as coral reefs are flattening. And what my husband, Jeremy Jackson, and I do is plot a sort of guesstimate of either biodiversity or ecosystem function as a function of reef degradation. So I hear you have places like the Northern Line Islands that Jen's going to talk about, which although there's been a certain amount of degradation, they still look like pretty nice reefs in some ways. And then out here, the Caribbean, places in Fiji and Kenya, where the extent of degradation has really declined. And so we can focus here on the richness of the coral associates. And probably out here, there's not been much of a decline. That's what we saw when we looked at the number of crab species in the Northern Line Islands. There was no difference between Kingman Atoll and Christmas Island. We're sort of in this region. But somewhere in here, somewhere when you start flattening and degrading reefs, you start really losing things. The problem is, we really don't, this was a guesstimate, we really don't know if it goes like this and then drops off suddenly, or if it's a steady decline, and if it drops off suddenly, where it drops off. These are all really important questions. But I think now with these standardized sampling methods and DNA analysis methods, we actually can answer that question. And so the bottom line is that there's a lot more at stake in corals and seaweeds, and I'd like to thank the 4 people, but who did a lot of them, essentially all the molecular work, like Teasley, Osrich, Sheeta, Matt LeRae, and Chris Meyer, and also my funders, and also Mark and others for hosting me here. Thanks very much.
There's time for questions if anyone has any questions.
Yes.
Nancy, I was thinking about what you said about the wrecking ball and the construction crane analogy of coral reefs. And in relationship to your artificial surfaces for settling, can you comment on the role that the timing plays? Like if you're going to have an early successional or a late successional community on these artificial setups you're going to get very different answers. And I would think that if you go later, you get a mature community, but then you don't have that wrecking ball on these artificial surfaces. So how do you take timing into account?
Well, one of the ways we've been doing that is to leave them out for different lengths of time.
Mm-hmm.
Obviously, we can't leave them out, well, we could leave them out for 20 years, I just won't figure out to analyze it. So we have left them out for different amounts of time. We haven't really done those. Some of the ones that Noah had put out are being picked up now. They've been out for 2 or 3 years. So we'll get a feeling in the next year or so exactly how much of a difference it makes. The idea is really if you do the same thing, you're never going to know exactly how many species there are or how the number of species you're losing. The more important question, or the more practical question, is what the pattern is with respect to different levels of reef degradation or different kinds of damages or different amounts, say, seaweed growing on reefs. So if you do the same thing, put these kinds of structures out or sample dead coral heads, that's one of the reasons we vote, because they actually come out relatively, to be honest, in terms of specific answer to your question, one way of answering is to say that the things that you find on dead coral heads, the things you find on artificial structures are actually very, very similar. So they're not wildly different. But they are, on some level, biosample. Well, any sample is a biosample. When you send someone like Gustav Pauli, who's the world's greatest vertebrate censuser, out to find everything he can on a reef, that's a biosample. He samples it a certain way. He's going to miss something. The different scientists are different. virtue of this is that it sort of standardizes the approach, so you don't have to be so worried about whether, my sampling is different from Gustav's or is different from Boucher's, and you get the same, so you can compare regions, you compare time, you can do this thing through time, you can see, we can do the same thing 10 years from now and see if things are different 10 years from now than they are now. Standardize the approach, and that's really the virtue. So as long as you're doing it the same way, the fact that in the case of the arms, they don't have biodiversity. I mean, they're made of PVC. There are all sorts of things that are not natural about them. But you do that, you do it with a natural surface, and you do it, whatever you do the same way in all these different places, and I think you get the answers that are most important. Yeah.
So based on the talk and going into the future, so if you recommend someone who is going to do biodiversity and try to see what's going on, if you recommend us as scientists going to the biodiversity hotspots, or if you recommend someone going to where there's already known information doing the future?
So the question is whether, I'm sorry, I didn't repeat that, hopefully people follow the question, the first one. The question is whether you should do this kind of work in biodiversity hotspots or places where we already know what's there. The answer, I guess, is both, and that's one of the great virtues of these methods, that we now actually can practically, logistically do both, whereas before it wasn't possible. One of the reasons we did this in Moorea was because they had spent 5 years trying to figure out every single invertebrate and fish species, and so we can take a look at the sequences that we get from this next generation sequencing and compare it to their barcode database, and that's how we know that we're picking up, we're missing a lot, but we're still getting a lot of correspondence between what we pick up and what they already know is there for 5 years. But obviously it's also really important to do it in a biodiversity hotspot, where it's one of the reasons we're doing it in Indonesia. Moorea actually was chosen because it's an relatively isolated island and relatively low diversity because it's in the Central Pacific. Indonesia is right in the heart of the Coral Triangle. It's mega diverse, and so it's really important to see what turns up in that kind of setting as well. So but you can do both now. Yeah.
Maybe following on from that, Nancy, how much of the global patterns do you think are slanted because so much of the work seems to have been done on the GBR Caribbean or a few Pacific islands, and there do seem to be these very swaths of coral reef areas like Indonesia, the Coral Triangle, where people just mostly haven't worked. So, any thoughts on whether you think that patterns are showing reflected in those places?
Well, I do know in Indonesia, we're in those 12 heads that the students picked up and analyzed, we got well over 400 decoupage. It was vastly more than anything we ever got. And that was really a quick and dirty sort of experiment last summer to see how it worked. I think what is one of the big unknown, some of the patterns are likely, part of the reason that, because we don't know much about the shape of that decline, that extinction curve is, this thing is because we have only done, we've done this in the Northern Line Islands. We've done it in probably in places in Western Atlantic. I don't have GBR here, but we've only done it in a few places until relatively recently. Actually, all the places we did it were out at this end of the spectrum because people were essentially ignoring these places that were in really good shape, because the reason they're in really good shape is they're so remote that they're really hard to get to, which is why people studies them. But I think a lot of the patterns will be, it'll fill in a lot of the shape of the curve. I don't know if we'll find something totally unexpected in terms of diversity patterns, so I suspect that some of the megadiversity spots are even more diverse than we realize, based on estimates from the more easy to sample things like fishes and large snails. We'll know, and I think in a couple of years, exactly how off we were. But the one thing where I really don't even feel like, I don't know how it's going to turn out at all. So I don't know how, if you put a bunch of, we do this in clusters, so we put 3 of these things in one place. They turn out that, and then to compare them 3 in another place, you get a sense of how much spatial variation there is. Within a small place, there's actually not that much variation. The arms are pretty similar. What we don't really have a sense of is how much how much variation there is from places that are, like how much difference is there from one place in Indonesia and another place? How localized is the diversity? The data we have so far suggests that it's really localized. We almost never find, we have a lot of overlap within sampling areas and almost essentially none between sampling areas. But we need to sample more places to know exactly how localized this localization is.
Can I follow along with that? You showed that, I think it was crab diversity as a function of Palmyra and other areas and along the heating gradient, said it wasn't different.
It wasn't a big, it's not
It's not much different, but there's a
small
huge,
sample size,
but
but not.
there's a huge difference in variance. In other words, in your less disturbed places, your error bars were huge. And in most disturbed places, they were small. And so do you get a lot of within-site variance in less disturbed places, and that would drive total diversity up?
I would say that those samples were so, this was done on a cruise, which is, as I generally probably taught, where we had 2 days per island or 3 days per island. So the sample sizes were, and I was doing it all by myself.
Yeah.
So the sample sizes are really small. I think that's a really interesting question, but we require, because you've got,
You've still got stuff to do.
I still got stuff to do. I think you just need a little, maybe 2 or 3 times the sampling incidence you have to even make a guess to the answer of the question. The question was whether places that are disturbed are more variable, less variable than places that are undisturbed. And it's reasonable, but I don't really feel like I have the data to answer it. Yeah.
The shape of those rarefaction curves speaks to the importance of rare species on reefs. Is there any evidence about where the biodiversity loss is taking place in that respect? I mean, is it the biodiversity lost by rare species dropping out of the community, or is it mostly concentrated in loss of common species?
Well, the question is where things are dropping out, whether it's rare things or common things. It is true that things are there are a lot of rare things. Usually about 30 to 40% of the sequences are singletons. That means the species only occurred as one individual. Just one of the reasons it makes it so hard to estimate diversity, because that increases the more rare things you have, the longer you have to sample to have any kind of clear understanding of the approximate number. Again, that's a question we haven't done it enough to really. We really spent these 5 years figuring out the methods, sort of trying in a number of places, working on this next-gen sequencing thing, which took years to get to work properly. And the question you're asking is like the question that Mark asked. I don't, logically I would say we'd be losing rare things rather than common things, but if it were something that was really common but dependent on living coral, then we probably could lose a quite common thing in that context. But I don't think we have, these data don't provide the answer to that either. Sorry, there are more questions than answers in this talk.
Yeah, when you look at the curve that you have from Line Islands out to the Western Atlantic, what would you expect based on island biogeography before there are any humans in a pristine state, that you have different expectations for your biodiversity? Just, extinction and origination rates are going to have different dynamics depending upon the size. So wouldn't you really want to be looking at changes relative to that baseline?
Oh yeah, that curve was, let me make sure I explain that properly. This wasn't a curve where we went from the Northern Line Islands to the Western Atlantic. It was more, it's a curve of quasi-pristine to wasteland. And so Western Atlantic is an example of something that's sort of at this end of the spectrum. But in any single setting, you can also, and that's one of the things we want to do now, is actually do controlled comparisons across gradients of just larger gradients than you can see in the Northern Line Islands, across major gradients of reef destruction, but within single places. So those, as you say, the history of the region, the native intact biodiversity, and all those other things are held constant. Indonesia would be a really good place to do that because they have beautiful reefs and complete vectors and so on. But the challenge is putting out the arms and finding them the following year. That's the biggest problem. The places that are wrecked usually have lots of foragers picking up strange objects. Makes it hard to do an experiment underwater. Yes?
I was wondering, with the next-gen sequencing, you're obtaining COX-1 or ribosome data to identify the individuals, or to identify species and getting an estimate of richness. Are you trying any way to complement that with data for evenness, or is that a later step down the road?
You can do even. Actually, there are evenness. When you do DNA barcoding, you can do evenness and total diversity easily, because you're doing it's a voucher-based approach, so that each individual is sequenced, and so we have the numbers of individuals that have those sequences. The challenge with next generation sequencing, when you put everything into a Waring blender and ground it up, is that it's very hard to tell the difference between one really big organism and 50 really small individuals. And so evenness is much harder to get with the next generation approach. On the other hand, it's much faster. So you just have to make a decision about how important these different kinds of questions are for what you're most interested in finding out about the system. So we have actually, the data that we published, the data on the barcoding is all, and the diversity of the crabs are all published. The next generation sequence data isn't. But in the published work, we talk about evenness as well as species.
Any more questions? One more? Thank you.